Thank you. Yeah, I really appreciate the support from ESPA to bring me here to present the work we have been doing together in Kenya. So this is part of a large project which was funded in 2011 to look at ecosystem services and disease, and it's being led by Melissa Leach from STEP Center in University of Sussex. So we have one case study in Kenya. The others are in different places, one in Senegal, another one in Ghana, and another one in Zimbabwe, but looking at different disease problems. So this one specifically in Kenya was looking at land use change, specifically irrigation, and it was being implemented by 4 institutions in partnership. That's ILRI, where I work with. We also have Kenya Medical Research Institute. We have the University of Nairobi, and Department of Veterinary Services. So for introduction, of course, we know climate change and also demographic changes are placing so much pressure on the existing resources. And so there is some sort of like food insecurity which needs to be met through expansion of agriculture. And in Eastern Africa, for example, the areas which are still available for agriculture are the dry areas, because by now, all the wet areas have been used for one thing or another in terms of irrigation, in terms of rain-fed agriculture. So to boost, of course, food production, there is so much emphasis on irrigation in these areas, but we know that irrigation in itself has so much impact on the environment. One thing, the standing water masses, which is created by bringing in water, provides good breeding grounds for mosquitoes, a large species of them. There is also development of a large peri-urban establishments or settlement schemes which bring people closer together with animals and mosquitoes and also rodents. But also, because we are clearing land for agriculture, we tend to change the diversity of hosts in those areas. So we have more of small ruminants than the large ruminants. And so those are deemed to be factors which might change the dynamics of disease transmission in that environment. So for the objective we had in this project, we wanted to study, of course, the impacts of irrigation on diversity of hosts and so biodiversity, but also to look at more specifically on the risk of vector-borne diseases. I must emphasize this project was funded, this specific case study was funded to look at Rift Valley fever as the case study disease. But along the way, we introduced other multiple pathogens because we came across so many literature publications saying you are better off looking at multiple diseases at the same time because the drivers of these diseases tend to be common, and so you get a good picture by comparing disease transmission for a number of pathogens. That picture there sort of like emphasizes the main problem we are dealing with. So it's, of course, presence of water. And we have been asking, why does the irrigation scheme allow water to stand, even when farming is not ongoing? And the main reason we found is that it's also a source of water for people in the area. So when farming is not ongoing, you still find water being pumped to the irrigation canals. This is the study area which we are working in. It's in the northeastern part of Kenya, somewhere there. Oh, maybe I should move a little bit. But you can see this is a map showing the irrigation scheme which is found in a very dry area where it's used for pastoralism. And the green thread here is the main river that serves water to that area. And so it's an arid and semi-arid area which under normal circumstances you wouldn't find water. But because of these irrigation schemes, you know, water has been brought closer to where people are living in. So we did various studies. One of them was to do an GIS analysis to look at land use patterns, changes in land use patterns over so many years. And also we did entomological and epidemiological surveys to, of course, compare areas with and without irrigation in terms of mosquito species and in some pathogens, which I'll be showing you in a short while. Those are the maps which we got for the period before irrigation was initiated. That's in 1975. and up to the more or less current, which is 2010, not so much current, but these are the clear pictures or the clear maps we could get. Sometimes because of the cloud cover in the region, it's difficult to get good maps that you can use for land use, land cover analysis. The other thing we did in field surveys, and we collected, of course, trapping. This is what we call CDC light trap for capturing mosquitoes. It's not specific for a given species, but it's more broad, and it tends to give you a good picture of the diversity of mosquitoes you would get. And we also did animal sampling, but also we got an ethical approval to collect samples from people. So we did a linked survey where we chose households, and within the same household, we are collecting samples from animals and also people within the same households. And the pathogens we were looking for include Rift Valley fever, Brucella, Coxiella burnetii. You know, this is a Q fever pathogen, and others. These are common. This can affect both human and livestock, but other specific ones for people and maybe other animals like equines or birds include West Nile virus, dengue, and Leptospira. So our analysis looked at, of course, I said earlier on, the GIS analysis to compare land use patterns. But for the entomology and serological data sets, we did a spatial analysis in R using the Bayesian approaches, which capture the spatial correlation in the data sets. And this is a method which has been developed recently by Rue and others from, I think, Norway, where you first develop a mesh for the study area, and you overlay your points so that the points are linked to the mesh, and you are then able to calibrate the distances between the points you are using for your analysis. Those are the first results which I'm showing. This is, of course, on land cover changes, and the main one which I want to emphasize, I hope it's clear, is the change in the size of irrigation area under cropland has really expanded as we expect. And also the area under urban and rural settlements also has expanded. But many other areas have gone down because of clearing of the land for irrigation. The other thing I want to show is the density of hosts. And the specific ones I want to show are the camels and sheep. These are small ruminants, goats and sheep. These ones have remained more or less positive, meaning, of course, either they have increased in a small extent, but the other large mammals, many of them has really gone down. And the other thing I want to emphasize for these wildlife and livestock densities is this is an analysis for the entire area. It wasn't possible to do specific areas like irrigated and non-irrigated because the counting was done in a much higher spatial scale compared to the entomological and epidemiological analysis, which I'll be showing you in a short while. So that's the results of the mosquito surveys. And first of all, there are 4 comparisons which we'll make here. I first want to highlight the top and the bottom panels. So for the top panel, it shows you the mosquito densities during the irrigation season. And of course, you can see most of them have very, very high densities compared to when there's no irrigation. But of more importance is to focus on the non-irrigated areas. You see, those also get affected, or they get high densities of mosquitoes when irrigation is ongoing because of spatial distribution of mosquitoes from the irrigated areas. But during the fallow period, you only see mosquitoes within the irrigated areas. So this, of course, confirms that irrigation helps to expand the mosquito season, especially in those places which are irrigated. We should also compare between villages and farms. Villages are settlements where these people are living, but farms are specific areas where farming is ongoing. And we found that there's a significantly higher densities of mosquitoes in the farms because that's where water is mostly available compared to the villages. Of course, we went ahead and did a statistical analysis to show that land use, basically irrigation and rain, has an effect on these populations. But, and we also separated all mosquitoes from the Rift Valley fever mosquito vectors because they usually require specific conditions for their development. That's flooding for an extended period of time. The posterior distribution of those parameters, mainly the rain. The rain is the dotted line for all mosquitoes, but irrigation is the more, in this graph of mine, it's a continuous line, but here it's showing its dotted line. But of course, it's to emphasize that, irrigation has a higher mean effect on the total population compared to rainfall. From the epidemiological analysis from livestock, we found that there isn't so much difference between irrigated and non-irrigated areas in the prevalences of these three pathogens, Rift Valley fever, Coxiella, and Brucella, mainly because livestock move up a lot in the area. They actually during the dry season they are crest within the irrigated areas, and vice versa. So you expect them to have acquired exposure for this range of pathogens in different areas. And so when we did the posterior distribution of the parameters, we didn't see any significant effect. But when you come to infection in people, because people are more or less settled, you find pastoralists in their areas and also human farmers in the farmlands, we found a pattern whereby for mosquito-borne infections, you have a higher risk of infection in the irrigated area. But for these other directly transmitted pathogens, mainly leptospirosis and brucellosis, which are usually the higher density of livestock you have, the higher chance that you'll get these infections, then we found them to be like they are more in the pastoral areas. This Coxiella is a funny, it's a result which we couldn't defend on either side because you can expect it to see more of it in the pastoral areas because it's found mainly in ruminants, goats, sheep. But also we find literature saying if you have a dusty area that has been denuded or cleared, then you have more wind dispersal of the pathogen, and so it's a high likelihood that you'll see this pathogen being more prevalent in an area that's prone to wind dispersal, which it's still, for now, I can't really place my hands on that because we didn't give much emphasis to it. So, but of course we expect to see mosquito-borne infection in an irrigated area, but in Eastern Africa, for example, Rift Valley fever up to now has been associated with El Nino phenomenon, high flooding, high rainfall periods. There's no literature up to now which shows Rift Valley fever with irrigation, and so maybe we are showing this as one of the evidence that if you put irrigation schemes in areas that are conducive for Rift Valley fever transmission, then there's a high likelihood of having that pathogen being sustained in the ecology. And this is my favorite slide, and I think it's the last results I'm showing. We went to the irrigated areas where you are finding very high incidences of West Nile. This is serological results. It's not really clinical cases. So we find high West Nile, dengue, and Rift Valley fever. But if you compared that result with what you see in hospitals, these are the hospital results, cases which are reported in hospitals and which are treated for, you find most hospitals are treating febrile illnesses for malaria or typhoid, or sometimes brucellosis. So in a way, there's a mismatch. Although these results cannot be compared directly, these are serological findings and these are clinical cases, but we can make an inference that maybe hospitals are really mistreating most of these cases in the community for malaria or typhoid. So that's another area which really requires some capacity building in terms of diagnosis in the local dispensaries. So for discussion, of course, we know irrigation increases food production, but it has a negative effect in terms of the services, in terms of making these multiple pathogens to be highly transmitted in that area. As I said earlier on, we wanted to use multiple pathogens because we can see a picture where one intervention or one driver or one factor could affect multiple pathogens in a given area, and that's what we are trying to report here. And we didn't find, yeah, thanks. On this biodiversity, we didn't find a good correlation because areas which are inhabited by pastoralists also have a higher density of wildlife compared to irrigated area. But if you look at that pattern of infections which we showed in that graph, there isn't a good correlation between biodiversity and prevalence of diseases. So I have a last slide here which we're asking ourselves what is the way forward, and one of them is we want to communicate these findings to mainly people in the area and also to decision makers to understand how irrigation or land use can have an impact on disease incidence, and mainly to emphasize this shift in terms of prevalence of diseases. But also on provision of health services, I showed that our hospitals are really doing a mistreatment or misdiagnosis of cases because to them, what they have come to know over these many years is malaria. And so when you get new infections like dengue, which is not prevalent in that area, they may be misdiagnosed for malaria. But the big thing which I'm still wondering on how to do is how to introduce land use management or to discuss land use management and water management in that area, because that's actually the key focus which sustains mosquito populations. But as I said earlier on, this water is also being used by communities for their own domestic needs. So there should be a way in which we can introduce piped water systems so that we do away with the standing water in the irrigation schemes. So, of course, I acknowledge ESPA and also the people I've been working with in this project in Kenya. Thank you.
Thanks for that, Ben. Could you go back to your slide with the disease risk with irrigated non-irrigated? That one right there. So there's an increased risk of the Rift Valley fever, dengue, but there's a decreased risk of the other two. Is that a correct interpretation?
Yeah, for these two, this is focusing on the irrigated area. So compared to pastoral area, there's a decrease in lepto in irrigated, and also a decrease in brucella because of the shift in the hosts. You get more of the small ruminants, which may not be a good reservoirs for some of these diseases.
And in terms of the impact of these different diseases on lifespans or healthcare costs, are they equivalent? Which ones are really difficult to treat versus easier to treat?
Yeah, that's a good question. Actually, the one that really gives us a big problem is brucella because it takes a very long time to treat. You need to go for multiple treatments over time. These other ones, and that's actually a good question because dengue, maybe dengue also, but West Nile and Rift, majority of cases appear as short-term illnesses which may come and go. But these other ones, the bacterial infections are quite sticky.
Thanks very much. I know you said you didn't get many conclusions about the. Thanks. I know you said you didn't get many conclusions about the links between biodiversity and health or transmission of these diseases, but I think you said when you introduced yourself that you had a hypothesis originally about that you wanted to test. So can you tell us more about what your hypothesis was?
Yeah, so the hypothesis was high biodiversity would regulate disease, and so you get less disease in areas with high biodiversity. So in this specific case study, we didn't see a direct connection, but I'm hoping that, once we synthesize data from different case studies, because I told you there's one in Zimbabwe looking at trypanosomosis, there's one in Ghana looking at Lassa, and one in Sierra Leone looking at, I forgot a bit. But I think once we synthesize all those studies, there may be some trend which emerges, but for our case study, we didn't see much linkages. Okay, yeah, the literature says if you have multiple hosts, then there's a high likelihood that you have a host which is a dead-end host for that pathogen. So in such a case, you don't have a spillover of infections to human population. So, yeah.
Hi, I just have a minor question, which was on a previous graph, you were showing that that there's a difference between the incidence of malaria. It's on the one with irrigation with farms and villages.
Yeah.
So back a bit further, and I think that it looked like there was. So if you can find the slide, sorry, keep going back.
Yeah.
Yeah, that one. It looks like that in the non-irrigated area or on farms, there was no incidence of or no vectors for two of the vectors that you've been testing. Yeah, whereas at the village level, you're finding that non-irrigated areas, there are still vectors. So I was just curious as to if you have thought about why at the village scale you're seeing vectors on non-irrigated areas but not at the farm scale.
Oh, yeah. I guess one of the things is some of these households keep animals within their enclosures. So at the backyard, you have animal pens being kept there. So they tend to be damp with some soils. And so I was thinking maybe that could be a way in which, mosquitoes may find some areas for breeding in those areas where animals are being kept. I think I need to tease out the specific species of mosquitoes which are found there compared to the farms. But what I know at the back of my mind now is the farm is very much good for Rift Valley fever vectors.
Okay.
Yeah.
Thank you very much, Bernard. Another round of applause for him.